Welcome back, everyone, to another explainer. Today we're looking at something you've probably heard about 1,000 times, but we are going to look at it through a completely different lens. We aren't just talking about DNA as your standard biological concept. No, we're going to examine it as an absolute masterpiece of structural engineering. We're looking at the literal nano-architecture of the blueprint of life. I want you to think of it less like a squishy biological molecule and more like a towering, perfectly calibrated skyscraper on a microscopic scale. Okay, let's dive right into this and take a look at our roadmap. We'll start with the blueprint breakthrough, zoom in on the chemical building blocks, and then look at the master architecture. From there, we'll map out the mathematical geometry, uncover the forces holding it all together, and finally look at dynamic DNA forms. Section 1. The blueprint breakthrough. Every great building starts with a breakthrough, right? And for DNA, it was just this incredible historical detective story. Just imagine trying to figure this out with mid-20th century technology. First, we had Rosalind Franklin and Maurice Wilkins. Their X-ray diffraction data, specifically that highly famous photo 51, proved there was a helical structure with regular repeating periodicity. Basically, it was a spiral. At the exact same time, Erwin Chargaff was figuring out the math, discovering strict rules regarding its base composition. So Watson and Crick didn't just discover the structure out of nowhere in a vacuum. They actually assembled these highly specific puzzle pieces provided by others to finally propose the iconic double-helix model back in 1953. The blueprint was finally visible to us. Section 2. Chemical building blocks.
So DNA is essentially a polymer, a really long chain of what we call deoxyribonucleotides. If you're constructing this DNA skyscraper, you need three main components for every single brick. Step 1 is the pentose sugar, Step 2 is the nitrogenous base, and Step 3 is the phosphate group. Now, these components don't just magically float together. Think of N-glycosidic bonds and ester bonds as the heavy-duty industrial steel rivets locking these three microscopic materials together into one sturdy, solid nucleotide.
And this brilliantly illustrates why DNA makes for a superior, incredibly stable building material. Just look at the sugar in DNA, 2-deoxy-beta-D-ribose. The key is literally right there in the name, deoxy. It completely lacks a hydroxyl group or an OH at the C2 position. It's just a simple hydrogen atom sitting there. Compare that to RNA, which retains that reactive hydroxyl group. Because RNA has that reactive oxygen, it's chemically unstable. DNA, missing that oxygen, is structurally sound and built for long-term data storage, which, is exactly why your genetic code doesn't just spontaneously degrade while you're sitting there watching this. To build our tower, we have two sizes of bricks serving as the actual letters of our genetic code. We have purines, which are adenine and guanine, and these have a larger double ring structure. Then we have pyrimidines, thymine, and cytosine, which have a smaller single ring structure. And remember how these attach to the sugar we were just talking about? The N-glycosidic bond links the carbon 1' of the sugar directly to the nitrogen 9 of the purines or the nitrogen 1 of the pyrimidines. It is a highly precise docking mechanism. Section 3. The master architecture. Let's move to and see how this actually builds into the standard physiological form, which we call bDNA. First off, it's a right-handed double helix, meaning the two chains coil around a central axis twisting to the right. Second, and this is absolutely vital, it has an antiparallel orientation. One strand runs in a 5' to 3' direction, while the other runs exactly opposite, 3' to 5', kind of like a two-way street. Finally, the structure plays incredibly cleverly with its environment. The sugar-phosphate backbone forms a hydrophilic or water-loving outer railing, while the hydrophobic water-fearing bases hide away on the inside. This naturally protects the actual data, the genetic letters, from the harsh, watery environment of the cell, while the tough backbone takes all the beating on the outside. Inside that hydrophobic core, the bases pair up in a very, very specific way. Adenine pairs exclusively with thymine using exactly 2 hydrogen bonds. Guanine pairs exclusively with cytosine using 3 hydrogen bonds. And hey, this perfectly explains Chargaff's rules from our earlier history section. Because A always pairs with T and G always pairs with C, in any double-stranded DNA molecule, the total number of purines is always going to equal the total number of pyrimidines. The math just perfectly balances the architecture. Section 4. The mathematical geometry. If you were to measure the diameter straight across the bDNA double helix, you would get a perfectly standardized 2.0 nanometers or 20 angstroms. Think about how incredible that is for a second. Because a double-ring purine always pairs with a single-ring pyrimidine, the width literally never bulges or pinches. a biological molecule having a perfectly standardized width across billions of base pairs. That is a mind-blowing feat of natural engineering. It's perfectly smooth and consistent all the way down the line.
Next up, we have the pitch. If you trace the spiral down, the exact distance it takes for the helix to make one complete, majestic 360-degree turn is exactly 3.4 nanometers.
So, the crucial point is, how many steps can you actually fit into a single floor of this spiral staircase? The answer is precisely 10.5 base pairs packed into every single complete rotation of this structure.
And if we do a little division to find the rise, which is the incredibly tiny vertical distance from one adjacent base pair step to the next, we get exactly 0.34 nanometers. It is a structure of absolute, undeniable mathematical precision. And it's this extreme precision that allows the reading mechanisms inside our cells to smoothly scan the data without making devastating errors.
But here's a really fascinating architectural quirk. Because the glycosidic bonds connecting the bases to the sugar backbone aren't exactly directly opposite each other, the backbones are not equally spaced. As the helix twists, this natural geometry creates alternating grooves. You get major grooves, which are wider and expose more chemical groups. You can kind of think of these as the access panels or maintenance shafts for the building, where protein crews can actually get in and read the blueprints. And alternating right alongside those, you get minor grooves, which are the narrower spaces along the helix.
Section 5. Forces holding it together.
Now, what's really interesting about this next part is, well, it's a massive physics problem we haven't even talked about yet. Those phosphate groups on the outer railing, they give DNA a highly concentrated, very strong negative charge. And we know that like charges repel. So with thousands of negative charges packed right next to each other, furiously repelling each other, why doesn't the entire helix just rip itself apart? The answer lies in three stabilizing forces working in perfect harmony. Think hydrogen bonds do all the heavy lifting? Actually, think again. Hydrogen bonds are relatively weak on their own. They mainly just provide the sequence specificity for pairing. The real hero, the primary pillar of stability here, is hydrophobic stacking. Those flat, hydrophobic bases stack tightly together like a roll of coins, using van der Waals forces to minimize their exposure to water. That provides massive core strength. And to solve our negative repulsion problem, ionic shielding. The cell cleverly uses positively charged magnesium cations and basic histone proteins to neutralize all those angry negative charges on the outside railing.
Section 6. Dynamic DNA forms.
This brings us to a concept called structural polymorphism, and it's going to completely shatter the illusion that DNA is just a rigid, unmoving statue. It's the phenomenal ability of the DNA molecule to dynamically adapt and physically shift its entire conformation. You see, the B-DNA we've been talking about is just one state. DNA can actually morph into other architectural styles entirely, like A-DNA or even Z-DNA, which literally twists entirely backwards into a left-handed helix. It's a dramatic structural shift.
So what actually causes this massive structural shift? The environment. The building literally changes shape based on cellular hydration levels, the local salt concentration, and even the specific base sequence itself. The architecture is responsive. It breathes. It reacts to the conditions of the cell around it in ways that are, frankly, just awe-inspiring.
Which leaves us with something to really think about. If the very blueprint of life, the fundamental molecule holding our genetic code, isn't just a static, boring ladder, but a highly dynamic, responsive, shape-shifting marvel of nanoarchitecture, what else about our biology is vastly more dynamic than we ever realized? Thanks so much for watching, and keep exploring.